Today's lesson on cellular transport. Each cell has a cell membrane, which is a skin for the cell that acts as a gatekeeper. We say that the cell membrane is semi-permeable, and that means it lets some materials through, but it doesn't let other materials through. Specifically, small molecules and nonpolar substances can go right through the cell membrane, right through the skin of the cell, and larger items and charged items like ions and polar molecules can't get through the cell membrane. The cell membrane is made up of two layers known as the lipid bilayer. You want to think of your cell as being double bagged, like at the grocery store when your groceries are put in one bag and another bag. That's kind of like the cell membrane. And the cell membrane is made up of something called a lipid bilayer. If you remember from biochemistry, a lipid is a fat, and a phospholipid is pictured on the bottom. And you can see the phospholipid has this head and it has two tails. The heads of a phospholipid love water, so they face towards water. Inside of the cell is water, and outside of the cell is mostly water. So all the heads face outside and they face inside. And the tails are hydrophobic, which means that they fear the water, so they face each other and they face away from the water. So that's what the structure of the cell membrane is. Now, materials can move in and out of the cell through either active or passive transport. We'll look at passive transport first. Passive transport uses no energy, and this is because substances tend to move on their own. They're always in motion. Particles are always in motion, moving and bumping into each other, and they tend to spread out on their own without any energy needed to be added. There are three types of passive transport, and we'll look at each. There's diffusion, facilitated diffusion, and osmosis. And then we'll look at active transport, which requires energy. It requires ATP. Before we talk about diffusion, let's clarify some terms. Concentration is the amount of substances in a given volume. So if you look at this picture on the bottom, there's two beakers. Each of them have 10 particles in them. However, the beaker on the left is really small and the beaker on the right is really big. The beaker on the left is more concentrated. There's more substance in a given volume. That is, the particles are more bunched together. It's more crowded, it's more concentrated, whereas the one on the right is less concentrated. Concentration gradient is what happens when one area has a higher concentration than another. And on their own, molecules are constantly hitting each other and they're trying to go down their concentration gradient. They want to go from high concentration to low concentration. Kind of like if you were all bunched together in the elevator and the elevator doors open, there's a lot of space. So you are going to move apart from all of your friends in the elevator and you're going to spread apart. You're going to move down your concentration gradient naturally without any energy needing to be added. So in simple diffusion, small nonpolar molecules go right through the lipid bilayer, they go right through the cell membrane, and they go from high concentration to low concentration. On the picture on the bottom left, you can see that there is a cell and there are all these little particles outside the cell in a very high concentration, whereas inside the cell, they're in low concentration. So what happens to these particles, since they're very small and they're not charged, is they're going to go down their concentration gradient. They're going to spread out and move into the cell so that at the end, we reach a state known as equilibrium, where the concentration of these molecules is the same outside the cell as it is inside the cell. Another demonstration of this is what happens when you take a lump of sugar, or in this case a cube of sugar, and you put it into water. The sugar molecules are all bunched up together right next to each other, and they start to hit each other and spread out. They go from where they are more concentrated in that clump to where they are less concentrated, which is the other parts of the beaker. And over time, the sugar is going to dissolve and it's going to spread out evenly throughout the entire beaker. That's called diffusion. No energy is required for diffusion. The sugar will just spread out on its own. The molecules will just come into the cell on their own. The second type of passive transport is facilitated diffusion. This is the same thing as diffusion. However, in facilitated diffusion, we get a little help. That's why it's called facilitated diffusion. And that help comes in the form of a channel or carrier protein. This purple structure right here is a channel protein. And what happens here is that ions and polar molecules need to get through the lipid bilayer, but they can't pass directly through here. They can't get right through the phospholipids. They need a channel. However, they're going to act much the way that they acted during diffusion. There's a high concentration of particles outside the cell, a low concentration inside the cell. The molecules are going to go down their concentration gradient and they're going to keep coming into the cell until they reach a state known as equilibrium. No energy is required for facilitated diffusion. The third type of passive transport is known as osmosis. Osmosis is really just the diffusion of water. When we're thinking about osmosis, this always involves a semi-permeable membrane, which again, it's a structure that it lets some things through, but not other things through. Kind of like a colander when you're making pasta and you're trying to drain it. The water is going to get through and the pasta is going to stay behind. In this case, these little molecules are going to stay behind. They can't pass through the semi-permeable membrane, which in this picture is indicated by this little red dotted line. So what happens is that water is going to go down its concentration gradient. It's going to go from where there's a lot of water to where there is less water. So if we look at the first beaker, on the left-hand side, there's a lot of water molecules. Everywhere there isn't a little purple dot is a water molecule. On the right-hand side, there is not so many water molecules. There's a lower concentration of water. So what happens in this situation without any energy being added is the water is going to come right through this semi-permeable membrane and it's going to move to the right-hand side of the beaker. The water level on the right-hand side is going to rise. And at the end of this, when we reach equilibrium, there's going to be an equal concentration on the left-hand side and an equal concentration on the right-hand side. This is known as osmosis. We're going to look at at several instances of osmosis next. In the first situation, and we're going to look at a red blood cell. We're going to look at what happens when we put the red blood cell in something known as a hypotonic solution. A hypotonic solution is a solution that has a lower concentration of solutes and a higher concentration of water than the cytoplasm of the cell. So if we look at this picture on the left-hand side, this is a red blood cell. Here's its outline. And it has 20% salt concentration inside the cell. It's in a hypotonic solution that has only 10% salt concentration. This means that there's a higher concentration of water outside the cell and a lower concentration of water inside the cell. So water wants to go down its concentration gradient because of osmosis, and water is going to rush into the red blood cell. That is not good news. So if you were to get an IV that was pure water, that would be really bad because water would just enter right into your red blood cells and they would swell and they would even burst, like you can see in this drawing, and you can see the swollen blood cells on the photo. So the result, water moves from the solution to the inside of the cell, cells swell and burst open. The second type of solution that you don't want is a hypertonic solution. A hypertonic solution has a higher concentration of solutes and a lower concentration of water than the cytoplasm of the cell. So in this scenario, our red blood cell has a 10% salt concentration, whereas the liquid is hypertonic. Hyper means more than. So it has a higher concentration of salt. It's 20% salt. And in this situation, there's more water molecules next to each other inside the cell, and there's less water molecules outside the cell. And what happens is the water is going to go down its concentration gradient. It's going to rush out of the cell. And as a result, the cell is going to shrink. These are plant cells that actually shrink because they are losing water. And the red blood cell here has done the same. It has lost its water, and that has caused it to shrink. This is because it was placed in a hypertonic solution. So the third type of solution is an isotonic solution. In an isotonic solution, the concentration of solutes in the solution is equal to the concentration of solutes in the cytoplasm. On our image, there's 10% salt concentration in the cell, 10% salt concentration out of the cell. So water is going to move into the cell and out of the cell at roughly the same rate. It's going to already be at equilibrium. As a result, the red blood cells will stay and keep their normal, healthy shape. This is the type of solution you would get if you went to the hospital to get an IV. You would get an isotonic solution. Okay, that was all passive transport. Active transport requires energy. During active transport, molecules are going to go against their concentration gradient. They're going to go from low concentration to high concentration, and active transport includes pumps and vesicles. Pumps. In active transport, carrier proteins require energy to pump substances against their concentration gradients. So if we look at the image on the bottom, what we can see here is that there is a low concentration inside the cell of these little green particles, and there's a high concentration of them outside the cell. So normally you would have these little particles move into the cell. But what's happening here is we are actively pumping these little molecules out of the cell. Maybe we just really don't want these little green molecules, and you're going to have to take energy because you're pushing these molecules against their concentration gradient. You're pushing them where they don't want to go. An example of this active transport is the sodium-potassium pump. And the sodium-potassium pump is a carrier protein that uses ATP to actively transport sodium ions out of the cell and potassium ions into the cell. Why does it do this? This prevents sodium ions from building up inside of cells, which could lead to osmosis, causing cells to swell or burst. A quick comparison. In diffusion, molecules are going from high concentration to low concentration right across the cell membrane. In facilitated diffusion, they're doing the same thing, but they're not going directly through the cell membrane. They're going through a channel. In active transport, particles are doing the opposite. They are going against their concentration gradient. They are going from where they are less concentrated to where they are more concentrated. Think of it as an elevator is already filled with people and you're trying to shove more people on it. It's going to take some ATP. It's going to take some energy to get those extra people to fit onto the elevator. The second type of active transport involves vesicles. Large substances cross the cell membrane in vesicles. In endocytosis, large substances move into cells by the means of a vesicle. In exocytosis, materials move out of the cell by means of a vesicle, and the contents of the vesicle are released to the outside of the cell. Here's an example of endocytosis. You'll actually have the cell membrane basically pinch in and surround molecules and engulf them and pull them into the cell. This is how some cells eat. This is called endocytosis. In the next example, in exocytosis, it's the opposite process where large molecules are put into a vesicle which fuse with the cell membrane and allow the substances to leave the cell. Thank you so much for listening. I hope that was helpful.